Saskatchewan Agriculture's webinar on the ins and outs of herbicides. My name is Matthew Struthers, and I'm the Crops Extension Specialist with Saskatchewan Agriculture in Moose Jaw, and I will be chairing today's event. We'll start today with a few housekeeping notes. If anybody has any questions during the presentation, you may type them into the question box in the bottom right-hand side of your control panel. We'll be sure to answer them at the end of the presentation. Keep in mind when asking a question, it'll only be sent to me and will always be anonymous. During the presentation, you will be muted so that there's no feedback or that will interfere with the audio. The webinar will be recorded and a special link will be emailed out to you later today. Our guest speaker today is Clark Brenzil, the Provincial Weed Control Specialist with Saskatchewan Agriculture. Clark is based in Regina and provides information and recommendations on weed control to producers, agronomists, government departments and agencies, as well as municipalities. He has experience in weed management with the crop protection industry, university research, and government. He has been with the Saskatchewan Agriculture in this position since 1998. As well as part of this role, he administers the Weed Control Act, raising awareness about invasive weeds and assisting municipalities and landowners in understanding their respective roles and responsibilities under the Act. So with that, Clark, whenever you're ready, take off with it.
Okay, thanks a lot, Matt. So we're going to talk about the ins and outs of herbicides today. This is going to be at a fairly high technical level, so get your learning caps on and settle in for a nice, fairly extensive presentation. And there's going to be a lot of details in here. Luckily, the session is being recorded so that you can go back and reference it again in the future. If you get to a point and you think, well, I forget what Clark said about such and such a herbicide. Again, we've got over 270 specific brands of herbicides in the Guide to Crop Protection, and they're increasing every year. So I'm not going to talk about too much about specific herbicides, but what I'll do is I'll talk about the generalities with respect to the particular groups and how they work on the target weeds, and maybe a couple things that are specific to that group as far as the way that they work and the way that we can make them work more effectively. So to start out, what's a herbicide? Essentially, it's a product used to control weeds. That's kind of the simplest definition of what a herbicide is. When you get right down to the technical side of things, it's a compound or organism, and it can be a bioherbicide, not just a chemical herbicide, that blocks a critical pathway in a plant that results in its death or suppression. And so that essentially means that it's a challenge for that plant. It's one of those environmental challenges, and it's something that puts a level of selection pressure on a population of organisms. So that's something to also kind of keep in mind when we're talking about herbicides, is that it's kind of like a natural disaster, and we're dealing with living things here, and living things are all designed to kind of cope with natural disasters and get around those. So that's where our resistance comes from. If we talk about selective versus non-selective herbicides, a selective herbicide is one that'll kill a target plant within other desirable plants of the same or similar type without harm to that desirable plant. And so we do have several of those, and oftentimes selective herbicides can be somewhat more prone to the development of resistance, particularly the metabolic type, which is a multi-gene type of approach to resistance. And what the weed is actually doing is it's making itself more like that desirable plant that herbicide has been designed to be to not impact. And so you have a shift in that weed's population more to that ability that crop has to get it out of the system. So that takes us to the idea of mode of action. or we can use the analogy of herbicide resistance group with that as well. And essentially, it's the critical pathway in the plant that that herbicide interferes with. And within all the herbicides out there, they're all they can all be accumulated into groups of similar chemistry. And what they'll do is attack the same target site within a plant. And there is going to be a bit of variability. So if you get resistance to one of those herbicides within that group, then you tend to get resistance to all of them. And as I say, there are exceptions to that rule now, and I believe that that's going to be it's related to two things, is that you'll get different mutations happening within the target species. So one mutation may result in resistance to a subcomponent of that group. So if we talk about FOPs and DIMs within group one, you may get one mutation that infers resistance to the FOPs. You may get another one that infers resistance to the DIMs, and you may get another one that infers resistance to all three of the groups, so DIMs, FOPs, and DENs. So um, it depends on the mutation, and in some cases, it also depends on how specific that herbicide is to the target site. And if that herbicide is very specific to the target site, then it's less inclined to develop resistance. That weed is less inclined to develop resistance to that particular herbicide. And so a good example of that is, and I'll talk about that in a little bit when we get to our graphics. A good example of that is clethodim versus post or tralcoxidum type herbicide. So the things like select versus post or achieve. You tend to be able to get resistance to post and achieve type herbicides. That select will actually control some of those populations. So that molecule within select is a little more specific to that target site. And so you have to get your mutation happening in exactly the right spot in order to get resistance to for clethodim. And generally, the herbicide groups are identified by either numbers, and that's the WSSA system, or Weed Science Society of America, or by letters, which is the Herbicide Resistance Action Committee. That is a collection of herbicide manufacturers and scientists out of the US. And then the Australians, of course, all also have their own unique lettering system that's different from the HRAC system. But they've recently started adopting the WSSA numbering system for resistance as well. So they're moving away from their lettering system more to the WSSA numbering system. So if we look at how herbicide works, we have to first look at what's going on in the plant that herbicide is going to attack. And essentially, we're looking at the enzyme system, and an enzyme system is essentially just a catalyst. It allows a reaction to take place very, very easily within the plant that wouldn't necessarily happen on its own. And so what you end up with is two components that come into a site, join up and move away as the essential component, or it could go the other way around. You could have a component come into a site, get cleaved in half, and go off and move away. And oftentimes where you're getting cleaving taking place is a detoxification type of approach, and where you're getting that building of things from smaller components. That's the building or protein production type of enzyme system that we're looking at. So once when we get into a herbicide, essentially what a herbicide does is it fits into that site and it blocks those components from going into that target site. And so it interferes with that pathway within the plant, and it can cause two things to happen. One, it can result in that component that's being built not being produced, and oftentimes this is the growth component type of herbicides that we're talking about. So those are ones that are often referred to as systemic, or essentially a toxic effect within the plant that kills the plant slowly over a period of time. And so you can see all the groups there on the right, and notice at the bottom there we've got a new one that's group 29. That's an industrial product that's called indaziflam, and essentially what it does is it inhibits cell wall synthesis and prevents the production or prevents the emergence of weeds from the soil. The other thing that can happen is that you can get one of those components that going into that system, especially in a detoxification type of system that backs up behind that process. And generally what will happen is that those are toxic to the membranes of the cell, and they're considered to be membrane disruption type herbicides. And those are the groups on the left there that are indicated. And it could be various different compounds, anything from, it could be free electrons that are attacking the membranes to ammonia to oxygen radicals and things like that. So we'll talk about those as we go along. And when I talked about I'm trying to get my pointer here. Oh, let's do it this way. Okay. So when I talked about the difference in selectivity between things like select and post, is that let's assume that for post resistance, the mutation happens out in this area of the enzyme, whereas select may be shorter than where that mutation is, and so it's more specific to that target site. And that's essentially what I'm referring to when I'm saying that something is more specific to the target site. And so that kind of gives you a bit of a visual representation of what I'm trying to get at there. Okay. There are some groups or modes of action that aren't really specific to one particular target site, or they're not really specific to a target site at all, but they may be targeting something that is an actual building block itself. So in the case of group 3s, what they're doing is they're binding to tubulin, and tubulin is the thing that allows cells to divide. So it's actually attacking that material directly and not allowing it to function properly and allow cell division to take place. And with group 4s, what they're doing is they're actually mimicking natural auxin pathways in the plant. And generally these things impact multiple targets in the plant. And essentially the effect on the plant is almost identical to the natural indole acetic acid within the plant. The only difference is the plant can't break that compound down like it does its natural auxin. And so as a result, you have an uncontrolled hormonal effect within the plant that continues to accumulate and ends up killing the plant at the end of the day. There's a couple other concepts that I want to talk about as far as how herbicides move within the plant. And so if we look at apoplastic movement, essentially this is movement that's moving in the xylem. So it's moving with the soil, water, or and moving passively in the plant via the evapotranspiration process. So as water gets pulled up in the plant and evaporated from the leaf surface, it draws more material up from the roots. And so from that perspective, you can have herbicide that's soil active and also systemic at the same time. What you'll notice though is that those herbicides, when they get drawn up into the plant, tend to get drawn up into the more mature leaves that are driving more of that evapotranspiration, and less so into the newer areas of growth. That's fine, because what happens is that at the end of the day, it ends up starving that plant for energy, and the plant will die as a result of that anyway. The other process that we look at is symplastic movement, and that's actually active movement within the plant. And so essentially the herbicide has to get into the plant cell, whether that's via the leaves or via the roots, and then it's actively moved in the same way as nutrients or sugar to those areas of active growth within the plant. And it's moving in the phloem typically, and moving through cells and between cells via the plasmodesmata between the cells and through cell walls that way. If we look at a foliar application in these kinds of systems, the apoplastic movement, you really don't get much movement from where the droplet lands. So because you may get a little bit of spread more from diffusion than anything else, or a little bit of movement as some of that water in the leaf moves towards the edge of the leaf. You may get some movement that way, but generally you get very little movement from the point of application. And in symplastic movement, what you'll find is that material goes into the cells of the leaf and gets moved actively into the phloem the same way that the sugar is getting moved out of the leaves. It's being produced by the leaves and moving actively to those points of active growth. And so in that symplastic type of system or the active movement system, that point of growth may vary depending on the time of year. And so in the early part of the year when the plants are, particularly with perennial plants, when those plants are actively moving material upward in the plant, that can mean that you get more accumulation of that material in the upper growing points and upper axils of the plant. Whereas in the fall, where those perennial plants may be putting material down to the root for storage, that movement is down towards the root. And so you'll find that that material will accumulate in some of those buds, new buds that are being formed down in the root, so that they're killed and not available for growth next year. Another concept that is, as far as the way that herbicides move, is that you've got acropedal, which means essentially upward in the plant. So it moves up to the top of the plant. And then you've also got basipedal, which means that material is moving down in the plant. So that's an example of, in perennial plants, typically your movement is acropedal in the spring and basipedal in the fall. So let's get into the specific herbicide groups and talk about how they move. And what I'll be doing is using some of those terminologies that we talked about earlier in the presentation to describe each of the herbicide groups as we go through. And we'll talk a little bit about what products fall into that group as well at the end of the day. We won't talk about them specifically, but we'll talk about what products fall into that group. So group 1 herbicides, they inhibit a pathway called acetylcoenzyme A, carboxylase. And essentially that pathway is responsible for the production of fatty acids in the plant. And that means that they get used to produce membranes and waxy substances like the cuticle on the leaves of the plant. So when you stop that production, the plant becomes more, it has difficulty producing cells for one, and it also has problems defending itself against drying out because it's got thinner wax layers. This moves actively to new areas of growth, so it gets applied to the leaves typically, and then moves to those growing points in the plant. Generally, there's very little to no soil activity with these products. Depending on the product, they can bind strongly to soil or not. And generally, they're broken down rapidly in soil via microbial degradation typically. In some cases, you're going to get hydrolysis taking place as well. And with the DIMs, they can be susceptible to UV radiation breakdown. And so that means that with the DIMs, you can have a daytime effect on their efficacy. Products applied in midday may not perform as well as products that are applied in the evening or the early morning with those DIM type herbicides. So that's something to kind of be aware of as far as a hint. And that's particularly important when things are being pushed to the edge a little bit. So somebody may be cutting the rate a little bit, and they may be, oh, well, we're going on a little bit on the late side. So that solar radiation and UV degradation is the thing that may be the straw that breaks the camel's back, so to speak. And typically these things get applied with an oil or solvent-based adjuvant, so things like a crop oil concentrate or something like a solvent like a merge type product. The clethodim products are a little bit different because they get applied with phosphate ester type of adjuvants like Amigo, and that is a little bit of a water modifier and a little bit of a soapy type product. So it's a little bit different approach to that, but generally you can have a bit of interchangeability with adjuvants within these products. So if we look at the herbicides that are out there, one thing to talk about is that there's selective and non-selective group 1 herbicides. And that seems like it's a bit of an odd concept because group 1 herbicides on their own only control grasses. But when we talk about selective and non-selective group 1s, the non-selective products are the ones that we use in broadleaf crops. And so what they do is they control all grasses, whereas the ones that are considered selective are the ones that we can apply in cereal crops. And so what you're doing is you're selectively controlling one type of grass within another type of grass. And so that's a little bit different type of selective activity than we normally think of when we talk about herbicides that way. So if we look at what products are out there, so we've got cladinafos in the FOPs, the cereal herbicides. So that's the things like Horizon. We've got 12 lookalike products there. The phenoxaprops or the Pumas, we've got 5 products in that active ingredient class. And that also includes products that might be combination products. So it may be that active ingredient combined with a broadleaf product or with another grass control product. So it's not just standalone product of that active ingredient, but also combination products. In the DIMs for the cereals, we've got trocloxidum, and there's 4 products there. And then with the DENs, we've got 1 active ingredient as well that is pinoxidin, and that there's 10 products now in that class. If we go to the broadleaf crops where we've got non-selective products, we've got quizalofop, which is originally Assure. There's 10 products in that class now. And then we've also got the DIMs there as well. So clethodim, so Select or Centurion. We've now got 14 products that have clethodim in the guide now. And then sethoxidum, there are 3 products, and essentially you've got Post, and you've got a couple of other products from BASF that are combined with Post, essentially in the package. So you can see that we've got a lot of copying going on in the Group 1 class. So it's there's a lot of pressure on this class with respect to resistance, and so it's something to kind of keep in mind that they're products that have worked very, very well in the past, but we are seeing increasing resistance to this class of herbicides evolving in Saskatchewan for sure. So if we look at what's going on in these with these herbicides, if we look down here at the base of the plant, you can start to see that after 4 days, you're getting a little bit of white showing up at the base of the plant. And just remember that with grasses, your growing point as a seedling is always down here. So if you're impacting the growing point, when your leaves get produced, you've got new material being produced down here that pushes the older material up. So this material up here is older than this material down here. So where you'll see symptoms occurring first is down here near the growing point. So you may see some whitening of tissues down at the base here. You may see some intervenal chlorosis, and you'll typically see that this new growing point has stopped progressing. And so after a little portion of time, you end up with something that's dead as a doornail. And when we're looking at symptomology, when we're looking at injury in the lab, in our crop protection lab where we do injury assessments, it's very important to provide samples that are in the process of dying, not things that are actually dead. because once things are dead, they're dead, and there's no real way to tell why they're dead. So try and look around for those things that have sublethal symptoms, like the ones that we're seeing in this picture. And then that's much easier for us to try and get a handle on what's going on with that population. And oftentimes you'll find that kind of at the edge of the field where there was a little bit of a miss or a sublethal dose that took place. So those are the places to look for clues as to what's going on in a particular field. One easy way to tell if your group 1 herbicide is going to work is the tug test. And essentially after about 5 to 7 days after treatment, you can tug on that newest leaf, and then that leaf will pop right out. And essentially what's going on down there is that, again, you're losing cell membrane integrity down there because of the group 1. And as a result, you're getting some of that cell material spilling out into that area around the growing point. And you can get mold growth and other organisms starting to attack that sugary type liquid down there. And essentially you've lost the integrity at the bottom of that leaf, and it just pops right out. So some symptoms of injury to plants that shouldn't get injured, things to look for with group 1s. Essentially what you'll see with group 1s that are selective group 1s that are being used in cereals is you'll get bands that occur on the plant, and generally these things are temporary injury. They're not something that will kill the target plant or the non-target plant outright. Essentially, it happens under very specific circumstances where you've got cool conditions, or you've got saturated soil conditions, maybe even really, really hot conditions where that plant is shut down and it's not really metabolizing the herbicide as well as it might be. And so you end up with a little bit of symptomology showing up during that time frame when the plant is unable to cope with it. Once that condition clears itself, then the plant is able to continue that again, continue its growth normally, and it just continues on. And you get a lot of frightened people when those things, these areas here, when they start to flop over and the whole field turns white, then the phones start ringing. But generally, if you've got green tissue coming down here below that white band, there'll be no impact on that plant. It'll continue growing, and there'll be no sustained problem with that plant or yield loss typically. So with group 2s, what they do is they're another enzyme system blocker of an essential component for growth, and they're blocking the production of branched chain amino acids, and that's for the production of proteins within the plant. Essentially, the symptoms you're going to get with these are deficiency symptoms. So you'll end up with yellowing in the growing point and things like that because the plant has kind of run out of that component that it needs for growth. If you've got soil residues of these things and you're looking at injury, what'll happen is that you'll get these plants emerging up to a certain point, and then once the seed stops supplying them with that essential component for growth, then they'll start to exhibit the injury symptoms, and the plant will start to go down. So we talked about it prevents plants from producing protein. Depending on the class of herbicide you're looking at, you've got selectivity on both grasses and broadleaves. And these things can be easily tweaked in the lab to give you different spectrums and different crop sensitivities and different weed sensitivities. So it's a very versatile group to be able to work with if you're a chemist with an ag chem company, to be able to create new unique products. Typically they're taken up by both roots and shoots, and they're moved in the symplast. So they're actively moved, they go into the cells, and then they move to where the target site is. There's little binding to the soil. They can be very mobile in the soil if they're somewhat persistent. And the range of persistence is very wide. So you can have some that are broken down very, very quickly, and then there are others that can last for years in the soil. So typically the ones that hang around for a long time are not broken down very well with microbes, but rely exclusively on hydrolysis and are impacted in that sense by soil pH to some degree as well. Whereas there's others that rely more on microbial breakdown, and there's some that have both of those processes taking place to allow breakdown. And essentially what hydrolysis is it's just a water molecule comes along and it cleaves that active ingredient molecule in half and deactivates it as a herbicide. So it allows it, it's a quick way to get things broken down, especially if early on in the season where you may not have a very robust microbial population at that point. So you may be able to get a little bit more breakdown taking place earlier on in the year with hydrolysis. So here's our product cross section. So we've got some of our older products like Muster. There's some with very few lookalikes. And then you get down to the products like thiophensulfuron and tribeneuron. And so thiophensulfuron goes under the brand name of Refine originally, and also is in Refine Extra, but also tribeneuron is in Refine Extra. And so that's why you've got so many products there in that using the tribeneuron active ingredient. If we go to the imidazolinones, there are things like imazamox that's in Solo and Viper and Altitude, and also imazapyr, which is Pursuit, imazamethabenz, which is Assert, and imazapyr, which is Aries and Arsenal. The imazapyr in Aries is very, very low, whereas in Arsenal, it's the main active ingredient. And if when you're looking at Arsenal, it's a bare ground product for industrial vegetation control, whereas Aries is selective for use in pulses. You'll also note that with the imazamox, we've got lots of products that are using that active ingredient now. If we go down the list, we've got our triazolopyrimidines. So those are the things from Corteva that are things like fluoroacylam and pyroxalam. So fluoroacylam active ingredient, we now have 30 products on the market that use that active ingredient. So that's a very prolific active ingredient now as well, very similar to tribeneuron. Simplicity is fairly specific to the Corteva products now, and there's some combination mixes with not just Simplicity, but with other combination products as well. If we go down to the sulfo-aminocarbonyl triazolinones, Everest is the proprietary product for flucarbazone, and there's 10 products now that utilize flucarbazone. And there's also thiocarbazone that was developed by Bayer. And again, there's 4 products there, but they're largely combination products as well, where it's combined with other, say, broadleaf products. And then we got propylcarbazone, which is Olympus. One thing to kind of keep in mind here is that imidazolinones are a bit unique in this set of compounds, in that where the other compounds tend to be impacted by pH from the perspective of they're bound more by high pH soils. Imidazolinones are the other way around. They're bound more by acid soils. And so that means that they break down slower in acid soils, whereas sulfonylureas will break down slower typically, especially the ones that are breaking down via hydrolysis, will break down a little slower in higher pH soils. And so there's some differences there. The triazolopyrimidines and the sulfo-aminocarbonyl triazolinones, those are affected by pH, but only at a point where that pH is so high that you'd never really find it in a natural soil system. And so you really don't have to worry too much about that. It's more organic matter that will impact how present these things are in the system. There is a little bit of binding to organic matter with these that can kind of allow them to, that organic matter will buffer some of the soil activity of these products a little bit as well. If we look at a low rate on these, we can see that we're getting a bit of impact in the main growing point here first, where we're getting yellowing taking place in the new growth versus the old green growth. Typically what we'll see with an application of group 2s is that older growth will remain viable and fine for a period of time until it gets to a point where it needs to repair itself. And then at that point, that's when that tissue starts going down. So that could be 2 or 3 weeks from the point of application until you get death of those older leaves that were existing at the time of application. Again, yellowing of the newer growth, the older growth looks fine. We also get into disruption of the growth patterns of the newer growth as well. So this is the material moving towards the growing point and stopping that ability of that new growth to proliferate. One thing to kind of note here is the coloration. People often get hung up on color when trying to diagnose group 2 injury. And yes, they do produce a color, but that's just a stress indicator. And you can get that red or purple color being produced for several different reasons that are outside of group 2 herbicide injury as well. So it's something to kind of be careful about over applying a concept. You want to make sure that you're seeing these other sort of symptoms showing up as well before you start diagnosing group 2 injury. Again, we've got sort of that cessation of new growth in there. The older growth looks fine. You've got the cessation of the newer growth right away, distortion of the new growth, and change in color there. Stinkweed's very, very susceptible. This is probably, maybe 1 or 2 days after, because it is so susceptible. So you can see the yellow transition right in the middle of the plant first, and the older leaves are fine. One thing that we do see with soil activity with group 2s, and we see this with pursuit in particular on buckwheat, and we call it praying buckwheat. And what happens is that these first 2 leaves here will stand straight up on end and come together at their tips. And so that's why we refer to it as praying buckwheat. With grasses, again, the growing point is affected, so you'll often see symptoms start to occur right here at the newest growth. Again, interveinal chlorosis. Your newer leaves will start to yellow off. Your older leaves will more or less stay fine until they need to repair themselves. And then you start getting into injury to things that should be tolerating the herbicide. In this case, it's one of the group 2s that is able to be applied to a cereal crop, but in this case, it was they were being exposed under very cold conditions, and so we ended up with an injury scenario. The plant on the right is the one that was outside of the spray zone, and the one on the little shorter stunted ones are the ones that were in the spray area. And so what we end up with is stunting of the leaf blade in particular. And so what we end up with is a change in the proportion between the size of the sheath and the size of the blade. So you can see all these little miniature blades up here at the end of all these sheaths. The other thing that we'll see is a loss of apical dominance with group 2 injury at the growing point. And so what that'll mean is that you'll get a proliferation of tillers occurring as well. So they're not really seeing it too much on this one yet, but you will see it in some cases where you get many, many, many tillers showing up on a plant, or branches in the case of broadleaf plants. And again, you're kind of getting that interveinal chlorosis taking place here as well. And so this is a situation where you've got injury where you shouldn't have had injury. In broadleaf plants, what we end up with is a stunting of the internode region. So this pea plant was likely exposed right about when it was about this big. So any of these internodes that are established already at the time of exposure aren't affected. But from that point onward, you get compression of those internodes. And so you end up with stunting near that top of the plant. The other thing you get in this chickpea plant here, this is a chickpea field that a producer applied pursuit to ahead of planting his chickpeas, which is not recommended, but not an uncommon practice. And he brought this plant into the lab wondering about the disease pressure that was on the plants. And I just happened to peek in the box and go, you put pursuit on that field, didn't you? How did you know? Well, all this branching here is how I know, is that this is the proliferation of branches. You've lost apical dominance in this plant and it's starting to push out branches from every single node at the bottom of that plant. In this pea plant, you can also see here that you've got new growth coming at all the lateral nodes on that stem. So you've got new growth at each one of these nodes, and that's another indicator that you've got group 2 injury and loss of apical dominance there. Group 3s, these are the ones we talked about that don't attack a particular enzyme system, but actually attack a functional component in the plant, which is the tubulin. And that tubulin produces microtubules that allow cell division to take place. And generally, this happens in the roots. You can get a little bit of stunting in the young shoot as well. The symptoms you'll see are stubby clubbed roots that have failed to elongate. And generally above ground symptoms are not really particularly distinct from a normal plant. If the plant happens to be able to escape that and get through that treated layer, other than the plant is not thrifty because it doesn't really have too much in the way of roots. This really doesn't move from where it's exposed in the plant. So the plant is emerging within that treated zone of the soil because it's a soil active product. And it's not really moving in the plant at all. It's actually just that where that plant is affected is where it's exposed. These things bind fairly tightly to soil, and so they tend not to move around a whole lot. And they break down via anaerobic microbial degradation. So if you've put some of these things on in the fall and then have puddles in the field in the spring, you'll notice that in those puddles you have weeds breaking away. And the reason behind that is because of the anaerobic microbial degradation that's taken place in those puddles as a result of that water sitting there. The other key thing for breakdown is volatilization. So they're highly vaporous, these products, and they're also very sensitive to sunlight. And so that's the reason why these products require incorporation, and failure to incorporate these products, particularly in the spring where you've got high light levels and you've got warming soils, that will reduce your activity of these products. So best application tip for these is apply them in the fall, even though there is that risk that if you've got puddles that you could get breakdown of it in those puddles. But when you see loss of activity, what you'll see is loss of broadleaf activity first, then you'll see loss of cool season activity, and then at very last, you'll see the warm season activity being lost. And typically that's not something that's very common because these things are very, very active on warm season grasses. So these are the kinds of symptoms that you would see with group 3 injury. So you've got your clubbed roots here that are failing to elongate, and essentially instead of the root kind of progressing here and tapering off into a tip, it's like somebody's taken a string and just pulled it right back up into that clubby tip, and that's kind of where it sits, and it doesn't really progress after that. And this is an example of above ground symptoms. So the ones on the left have been affected by the herbicide. The one on the right has escaped and it's doing fine. The other thing we can see is, so the one on the right has been affected by the herbicide, the one on the left hasn't. And essentially you're just looking at a level of thriftiness that is the difference between the treated and the untreated. Group 4s, so these are the synthetic auxins. Um, Essentially, they're mimicking the natural indole acetic acid in the plant, but the susceptible plants can't metabolize it. It's a multiple target site type activity within the plant, and so that means that generally it's pretty robust with respect to resistance. These move actively in the plant via the symplast. So normally the indole acetic acid would be produced in the growing point and move downward through the plant. And so these things will do the same thing. They'll move through the plant that way. Generally, most of these are not bound very tightly to soil and so can be somewhat mobile, especially if they're persistent again. They're broken down largely by soil microbes, and they can have very short persistence in the soil, or they can have very long persistence in the soil. So an example of very short would be something like 2,4-D or MCPA, and very long would be something like picloram or aminocyclopyrochloric, where it takes a long time to break those things down. And in those situations where those products get used, that's a desirable characteristic. And these things can be absorbed by both roots and shoots. So because they're auxin mimics, dicots are the most susceptible. And so what you'll end up with is a concept called epinasty, which is the twisting of stems and petioles and cupping and curling of leaves. You'll get swelling of the stem, and right particularly at the lower stem, upper root kind of transition area. And that's where all the action happens as far as when it kills the plant. All of that twisting and curling and stuff is all just decoration to make us get a nice warm feeling in our belly that the product is working. But where the action really takes place is where you get callus buildup of undifferentiated cells at that lower stem, upper root area. And essentially what you end up with is that plant gets separated top from bottom by that excess growth right at that point there. And so one of the things that you will see on occasion as a result of that is that you'll get a splitting of the stem in those areas. Because indole acetic acid allows plants to elongate, you may also get exaggerated elongation within plants as well. You'll get chlorosis of the growing point because you're getting multiple target sites being impacted, and because you're cutting off the water transport from the roots, you'll end up getting wilting taking place. We talked about dicots being the most susceptible, but grasses are not immune to the impacts of that crushing effect that you get from the development of that callus tissue at that base of the plant. It's just their physiology is such that they tolerate that crushing a little bit more. And the symptom you'll get within a grass plant that is being impacted by a group 4 herbicide is that you'll end up with a plant that looks like it's under drought stress, even though there's lots of water around. And so you'll end up with thickening of cuticles, and you'll end up with the stunting taking place. You'll end up with particularly flag leaves that are shorter, because that plant is trying to protect itself from drying out. And so you can get very similar symptoms to drought symptoms when you have grasses that are impacted by group 4 herbicides. So the ones that we have in the different subclasses within the group 4 herbicides, so we've got our phenoxycarboxylic acids. So that's things like 2,4-D, 2,4-DB, MCPA, MCPB, and 2,4-DP, which is dichloropropyl, and MCPP, which is mecoprop. One of the things about 2,4-DB and MCPB is that they are proto-herbicides, and that in and of themselves, they don't have any herbicidal activity, But what happens is that when they get into a susceptible plant, they get converted into those other compounds. So 2,4-D will get converted to 2,4-D within a susceptible plant. And MCPB will get converted to MCPA within a susceptible plant. And generally, the way that these get used is that legumes don't have that mechanism to convert those things to the actual herbicidal component. And so you get tolerance in the legume family to these proto herbicides. And so that's the benefit of the butyric component of the salts in this case. In the benzoic acid class, we've got dicamba, and we've got quinclorac in the quinoline carboxylic acid class. In the pyridines, we've got things like aminopyrrolide, which is in range and pasture products, Restore and Reclaim, and in our IVM products, Milestone and Clearview. Clopyrrolide, originally sold as Lontrell. We now have 18 products, including generics and combination products. Fluroxypyr is a very prolific active ingredient now because of its activity on kochia and cleavers in particular. And we've got 45 products out there now that have fluroxypyr in them. And so that's something that is putting a lot of pressure on that active ingredient with respect to resistance right now. So something to kind of keep in mind there. Haloxyphen, it's also branded as Arlex or Aralex. That's in things like Paradigm and Pixaro, and now we have a few more products coming on the market like Prospect and things like that. There's 8 products in that class now, and I think I just saw another one here in kind of a newsreel that that there's another one that's essentially a Pixaro with clopyrrolide in it that's just been released by Corteva. Picloram is again, it's in range and pasture and IVM uses. And then triclopyr is typically used for brush control. And it's something that's very active on woody species. And then we've got a relatively new player in the pyrimidine carboxylic acid class. That's aminocyclopyrochlor, that's Navius, and now it's called Navius Flex. There's a new formulation out there. And so these are the impacts that you see on broadleaves. So you can see where there's, you've got elongation of these leaves, and you've got curling and cupping going on, failure to produce some of the lateral growth in these, the dandelions here. You've got yellowing of the growing point. The other thing to notice is this puckering that takes place on susceptible plants, where it's almost like the veins stop growing and the material inside doesn't stop growing. So you end up with it puckering upward or downward, and similar kind of, it's a similar kind of effect to what causes cupping in the plants as well. And so that's what we've got here is we've got cupping and curling taking place. Some cup backwards, some cup frontwards. We've got stem curving, and so the plant kind of loses its sense of up and down. This is the callus type response that I indicated was the business end of the group 4s. So that this is from a sublethal drift, and so you're getting a lot of this callus being built up on this plant without actually killing it right away. So it's been a kind of a slow, nasty progression in this case. And again, here's some callus being built up in the top part of the plant, and you can get it kind of mimicking staghead. And again, it's kind of a hormonal response. The other thing that you will see with particularly with things in the mustard family is that oftentimes you'll get kind of grooves that run up and down the stem where it almost looks like the stem wants to split long ways, and so you'll get one spot in the middle of the stem that is pushed inward, and then you get other bits that are still sticking outward. So we talked about grasses being tolerant but not immune, and so where grasses can sometimes be really susceptible is in the early stage of growth, just when that the seed head is being formed. And so that's why we've got a wait period on 2,4-D until we get to the 4-leaf stage. And then at that point, that head has already been formed down at the growing point. And so you'd end up with no more risk of that head being injured. The other symptoms we can see often are things like pigtailing of flag leaves. Oftentimes you'll get heads that get stuck in the boot and don't emerge properly, so that boot gets compressed up here, and so the head starts erupting out the side, and so you end up with head kinking as a result of that as well. And so the other thing you can get if you apply that herbicide too early in the growth stages is that you can get floret abortion as well. And so you'll end up with missing florets on the Reiki. And you can also get, this as a result of the head getting stuck in the boot as it emerges. And so you end up with the kinking of these heads, and the combination of that and the loss of florets can result in yield loss. So yeah, 2,4-D, the timing is that four-leaf stage. Early tillering is typically what's referred to. And for MCPA, because it's a little less harsh, you can get, especially if you go with lighter rates, you can get down to that 2-leaf stage. But if you go to 1-leaf stage with MCPA, you can still even get this kind of injury taking place as well, particularly at higher rates. Groups 5, 6, and 7 are all photosystem 2 inhibitors, and essentially they block electron transport. So in this case, you get free electrons flying around in cells, and they start attacking things that they don't like to be free. They like to be attached to things, so they tend to attack cell membranes, and whether that be the membranes on the outside of the cell or on the organelles within the inside of the plant, it doesn't really matter to them. They'll just attack any of those membranes. These things only move upward in the plant, and they move through acropedal or passive movement when they're applied to the soil. At low rates, you can get blotchy intervenoclerosis on leaves. Typically, though, what you'll see is especially if you have sublethal doses and you've got soil exposure or soil activity taking place, you'll find that you'll get a halo on the outside of the leaf starting to form with the symptoms there. And that's because all that material is moving out to the edge of the leaf with the movement of the water, and it's concentrating out there to the point where it becomes toxic. So groups 5 and 7, they're low to moderate binding in the soil, and they are available for root uptake. So that's where you can get that movement from soil to foliage. But group 6s are bound very tightly to soil, and so they're not available for root uptake. So they're only foliar herbicides. And so if we look at the various groups and the products within that group, those groups that are relevant to Saskatchewan, you've got metribuzin, which is in 8 products now and originally sold under Sencor. Hexazinone is Velpar, typically used in forage crops or in IVM type of applications. Atrazine used in corn type scenarios. And there's a couple of products there, including Atrix and things like Primextra 2 Magnum. Linuron, Lorox, or Linuron is the only active ingredient in group 7 really that we have access to. And Linuron is soon to be deregistered. So that one, group 7 is pretty much going the way of the dodo essentially across Canada. The group 6s, we've got bromoxynol, which is kind of a foundation compound for a lot of products, and originally sold on its own as partner. but now we find it in 30 products. And bentazone is the active ingredient originally in Bazagran, and it's now found in about 12 products out there. The symptoms that we see, this is the yellow blotchy thing, and essentially these blotches are all associated with the droplets as they land on the leaf, and that's a sublethal dose that we're looking at there. So it's not something that's going to progress rapidly to this. Sunflowers are very, very sensitive to the photosystem 2 inhibitors, and you'll get very rapid necrosis taking place at very low rates. And eventually, buckwheat is also very susceptible to things like bromoxynil, and it gets to the very necrotic stage in a very quick order. So you're looking at 4 days for total necrosis here. One thing that you can get with these products is shadowing that takes place. So because they're contact and they typically don't move around a whole lot, this green area here, you can see the sun shadow from the leaf above it. And so that leaf probably protected this leaf down here from exposure to the herbicide as well. And so you can see that you've got necrosis between the veins taking place in the areas where it was exposed. And it can be kind of inconsistent, but as long as you get enough of that leaf treated that that the veins start to be impacted, then you can get those leaves going down fairly consistently. One of the challenges we have with this class of herbicides is application to tolerant crops under very hot conditions. And so things like bromoxynil applied at very warm conditions, they act more quickly than the plant can actually cope with them and metabolize them. And so as a result, you end up with injury occurring to that plant that normally would be tolerant. And similar here with this oat plant, what's going on here is that the herbicide got applied and it got moved very rapidly to the tip of the leaf, again moving with that water in the plant and concentrating in the tip faster than that plant could cope with it, and so you ended up with some leaf tip burn as well. We also have situations where we get soil uptake, particularly if our plants are shallow seeded in light soils. We can get metribuzin moving down into that rooting zone, and then the top of the plant turns kind of this pale yellow color as a result. Group 8, these are lipid synthesis inhibitors, and that you'll probably think that that sounds familiar, and that's the same type of compounds that are impacted by the group 1s, but the group 8s use a different pathway, and it's not really as clear about how they impact the plant. And essentially what's going on is that those seedlings fail to emerge because it's a soil active product, and it's because they fail to form a cuticle layer, and you end up with poor membrane production. They're soil active. They're absorbed primarily by the coleoptile in grass and the hypocal arch and broadleaves, and there's actually very little uptake via the roots. It's mostly through those leaf organelles that are moving through that treated layer in the soil. And it moves very little from the point of uptake. If there is any movement, it's likely symplastic because it's moving towards that growing point. And you can have short to moderate persistence in the soil. And one situation that occurs with things like EPTC or EPTAM is that you have this concept, in particularly in Eastern Canada, that's called history soils. And what happens there is that the microbes become very well adapted at breaking that material down in the soil. And so rather than that product lasting 4 to 6 weeks in the soil, it only lasts for a few days, and you lose all of your weed control activity as a result. They can be volatile when they're combined with water in the soil, and so that's why they require incorporation. And if they don't get incorporated properly, they can gas off, and so that's why that's fairly important. To dry soil, they bind very, very strongly. The two products that are relevant to Western Canada are EPTC, so Eptam, and Triolate that's in Avadex and Fortress. So they're the only actives in this group, and Triolate only impacts wild oats and in a variety of crops. EPTC will impact both broadleaf and grasses. Typically, it's more active on warm season grasses than it is cool season grasses, but will impact cool season grasses as well, and it impacts a variety of broadleaf species. Symptoms that you get from injury in this case, so with corn, so if you put Eptam on without the corn safener that would be in its sister product Eradicane, you can get injury to corn, and so you end up with things like buggy whipping taking place, where the tip of the leaf gets stuck in the sheath of the previous leaf. You end up with and so that's what we've got going on here. You end up with distortion of the new growth. You can see there's a tassel kind of wanting to kind of come out here, so it's kind of premature maturation. With the broadleaf plants, you can get failure of these edges of the leaves to progress, and so you end up with that kind of lack of production of the side of the leaf and broadleaf species. Group 9, the only compound in that group is glyphosate, and there has been research that's been done on different compounds within that group, and primarily by Monsanto when they were first developing glyphosate. But glyphosate seems to be the one that is the most effective and the most versatile product that can be produced. So really anything that is a sister product is really not going to live up to the expectations of glyphosate, really. It inhibits the EPSP synthase pathway, which also produces branched chain amino acids. And you'll think, I've heard that before, and you've also heard it before with group 2, but it's a different set of amino acids that it's inhibiting. And so it's also inhibiting a different set of proteins. But what it does mean is that the symptoms of glyphosate injury are very similar to the symptoms of group 2 herbicides. And so there are some very subtle differences that you may be able to distinguish the injury of the two from one another. And largely that's due to the fact that glyphosate really only remains active in the plant for about 24 to 48 hours before it's bound up by inactive material within the plant. Whereas group 2s can persist in the plant and in the soil around the plant and continue to re-injure that plant over and over and over again. And so if you've got symptoms that are continuing to progress over and over and over again and not recovering, then you're likely looking at something like a group 2 versus a glyphosate injury if there's any question about that. So growth stops right after application. Your immature leaves in the growing point are affected first in about 5 to 7 days, and then again the older leaves go down as they fail to repair themselves. Similar to group 2s, the sublethal effects are yellowing into new growth, and that's because it's a deficiency of those essential amino acids. You get stunting and miniaturization, loss of apical dominance, so eruption of the lateral buds again, and compression of the newest inner nodes. Glyphosate moves very actively in the symplast and moves towards, again, what's the most active growing points in the plant. And again, so in the spring, you're looking at the top growth, which is the most actively growing. And in the fall, you're looking at the roots of perennials are the most actively growing. And so that's why timing of the application of glyphosate for the control of perennial plants is so important. Application in the fall when they're putting away material for storage for the following year is the key time to really apply glyphosate to make sure that you get good activity out of it on perennial weeds. It's bound strongly to soil, so that means that there's no organic or there's no soil activity. It's bound to organic matter and clay. It's degraded by soil microbes in the soil, and it's moderately persistent in soil. And a lot of people think that because it's not impacting the plant, that it breaks down very quickly, but it actually has a half-life of about 45 to 50 days in the soil. So it can last for a fairly significant amount of time in the soil, but because it's not really going anywhere, it's not really having an impact on the crop per se. And there's 29 products that contain glyphosate in the guide in 2021. So again, similar symptoms, 4 days. Again, stinkweed is very susceptible. Again, the coloration thing that we talked about earlier, where you can get that sort of flash of red going on, but don't get deceived, because remember what I talked about before is that you can get those flashes of red and purple for other reasons other than just glyphosate injury or group 2 injury. And in this case here, the key is this pea plant that looks very, very healthy. It's not being impacted, and the lamb's quarters back here in the back look very healthy. These buckwheat plants look very healthy, and so that's something specific to this brassica species that is causing the coloration change within that plant. So it could be that this is a pea field that has had an IMI applied to it, and that's what's going on there, but it's definitely not glyphosate. Grasses are very susceptible, so even at a low rate, they go down very, very quickly. And again, you're getting that coloration showing up in grass stands as well. Generally, though, you end up with dead is dead, and so you may or may not get that coloration change. One thing to kind of keep in mind is that there are other things that can mimic glyphosate injury in some species, so like flax. Yellowing at the top, there are several things that can cause yellowing of the top growth of flax, and in this case there's some clues in the background of the picture as to what might be causing that. In this picture, it looks like the soil is fairly wet within the row, and so you could get wet feet causing this yellowing situation. Aster yellows can also cause yellowing of the growing point as well. The impact on, and so this is on the right, this is an image that is consistent with glyphosate injury. And so what you've got is you're looking at miniaturization here of some of the new leaf growth, and a very sudden termination of the growth of that growing point. So it's essentially stopped, and it's a bright yellow, and it's just stopped dead in its tracks. So it's not really continuing to grow with yellow coloration. This is an ash tree on the left, and this ash tree was treated the previous fall with glyphosate. And so what happens is that when you see this, what's referred to as witch's broom type growth on trees, typically that's from an exposure the previous year, because trees are really good at packing away glyphosate in their roots and then putting it up into the new growth in the spring and causing the miniaturization of new growth, proliferation of branches and things like that around those nodes. So that's a pretty typical symptom of glyphosate injury on woody growth. And sometimes we do it to ourselves. So these are potatoes that were treated prior to harvest with glyphosate, and they ended up in somebody's potato storage house, and they started growing eyes that were all stunted in that. So it raised a real big flag right away with the folks that had bought the potatoes. And so there was a little bit of liability in this case, let's put it that way. Group 10, glutamine synthase inhibitors. So that's essentially glufosinate is the only one that's really relevant to Western Canada. There's a couple of things that happen with glufosinate and glutamine synthesis inhibition, is that not only is it inhibiting the production of those essential amino acids, glutamine and glutamate and others, but there's also a secondary effect that ends up being the primary effect in a lot of cases with Liberty application, is that you get an accumulation of ammonia within the cell, and that attacks the cell membranes and results in rapid necrosis. And so you can get both things happening in the plant at the same time to some degree. and the amount of one may dictate the amount of the other. So if you get very rapid dry down of the tissues after exposure, then that minimizes the amount of movement of that material out into the rest of the plant. Or whereas if you get a bit slower activity of that cell rupture, you can get more of that material moving into the rest of the plant. And the symptom that you'll get with that, with the glutamine and glutamate reduction, is kind of like a bleaching effect. And that's something that is a bit unique about the symptomology with glufosinate injury, is that even the desiccated tissues look whiter than the tissues that are desiccated with some of the other membrane disrupting compounds. It could be moved via the Symplast actively, but again, it can be self-limiting because of rapid necrosis of the leaves. It's weakly bound to soil. It's broken down rapidly by microbes in the soil, and so there's limited soil activity as a result of that. And this is what the injury would look like with the Liberty. You've got that lighter colored necrosis of the leaves. You've also got a little bit of movement into the growing point here. You've got the necrosis taking place on the sunflowers out here, but you're getting a little bit of movement into the main growing points of the plant as well. You often get spotting that is consistent with the droplet deposition on a leaf, and so that's something to kind of keep an eye out for. Group 13s, there's only one active ingredient, clomazone, in this group, and that's, I think, in total, there's only one active ingredient in that group, and what it does is it inhibits DOX P in the plant, and what that is, that's pigment production. So it reduces the production of carotene and chlorophyll in the plant, and you end up with tissues that turn white. It also impacts gibberellin synthesis, and that can result in some dwarfism or stunting. This stuff moves only upward in the plant through the xylem. It doesn't move downward via the Symplast. So it's a soil active product, and it's only through root uptake. It's moderately bound to soil, and it has short to moderate persistence in the soil and broken down by microbes. So susceptible plants typically emerge from the soil white and turn necrotic, and they die shortly afterwards. And you'll get bleaching on plants that are exposed after they emerge and are growing in the area. One of the things that is different about this product is that unless it's formulated properly in a microemulsion type of formulation or an encapsulated formulation, it can be very volatile in that you can get this stuff moving like a quarter mile if it's not formulated properly. And you end up with symptoms like this showing up on things that are downwind, so to speak. But the new formulations from FMC with Command is very they're not very volatile at all, and so you end up without that problem of volatility and it moving across the landscape. So these are the kinds of symptoms that you would see if you get exposure late in the growth period. So you get that bleaching effect. Group 14s are PPO inhibitors or protox inhibitors, and they inhibit protoporphyrogen oxidase, and essentially that's a long involved pathway that eventually results in what's referred to singlet oxygens that are highly charged, and so as a result they end up attacking membranes as well. They don't like to exist on their own, and so these things will largely move through acropedal movement in plants, but they can move in the phloem as well. The challenge with their movement in the phloem is that very rapid necrosis that occurs in the leaf tissue. particularly when they're applied directly to the leaves. And so what happens is that leaf tissue dies so quickly that you don't get movement in the phloem downward in the plant. And so it's largely just a contact type application. The soil active products, they move upward in the plant typically, but not very much downward. It's very short to relatively long persistence. So things like sulfentrazone can hang around for a fair amount of time, but you've got other things like carfentrazone that have a 1-day half-life. So it's right across the spectrum, and they're broken down via microbes, and they're lightly bound to soil. So we've got a bunch of subgroups within group 14 as well. So we've got the diphenyl ethers, which is the oldest class within the group 14s, and that's things like Reflex or Flex and Blazer, so aceflorofin and fomesafen. And then you've got flumioxazin, which is in its own subcategory, and I'm not even going to try and pronounce the name of that class. Pyroflufen has its own class. There are other things in those classes, but they're really not relevant to Western Canada right now. Same with saflufenicil, which is heat. So, and then there's a new class that is just under development by BASF, and it's probably getting close to the market here pretty shortly. So trifludimoxazin, and that's in the triazonone class of the group 14s. That'll be under the name of Viroxor or Volcaris. Viroxor is a combination product with saflufenicil as well. And then you also have your triazolanones, which are carfentrazone and sulfentrazone. So this is the kind of injury that you get from fomesafen when it's applied to foliage. Essentially it's a contact herbicide, and that bronzing appearance is very typical of the injury symptoms that you get. And this is on a soybean leaf, which is supposed to be tolerant. The weeds themselves, they'll essentially desiccate. But the injury on soybeans can be this kind of bronzy appearance, and you can see a leaf back here in the background that are showing that bronzing type of symptom as well. The soil active products, so things like sulfentrazone, they move upward in the plant via the water transport vessels. And so if they're doing that during the daytime, they get activated right within those veins. And so you can end up with a symptom that is venal chlorosis versus the intervenal chlorosis. You can also get, so you're seeing that both in this sunflower plant here and in these pea plants, where the veins are the first things to get impacted. And then that symptom spreads out from those veins into the rest of the plant. You can also get that situation where it gets moved up at a sublethal dose to the edge of the leaf and concentrates at the edge of the leaf, very similar to the other membrane disruptors that are active in soil as well. So you can get both kinds of injury kind of showing up and taking place. The other thing that you can get is with the soil actives is some splash occurring on leaves where it's on the soil, it gets splashed up onto the leaf and gets impacted that way. You can get a situation that occurs specifically under very saturated soil conditions and very sunny conditions under those saturated soil conditions. So in this case here, what we've got is that the sulfentrazone is free around the base of the plant as that leaf is being formed. And so as that leaf gets pushed up out of the area where it's produced, during the dark period, it gets to be able to push up material that's not really impacted. But during daylight hours, when you get it pushed up, you get this band that this is impacted and necrotic as a result. In the dark period, the plant has time to kind of metabolize it and deal with it and break it down. But in the daylight hours, it gets impacted right away. And so you end up with that banding type situation that can resemble heat banding to some degree. And you can kind of see the light colored bands up here as well on these leaves, where you can see this has progressed for several days in a row. So that's the kind of symptom that you'll see on cereal crops. If there's, again, if it's really, really soaking wet and there's a residue of sulfentrazone in the soil. Group 15, again, are long chain fatty acid. And again, these are cell membrane things, and again, not group 1 related. So the similar type of activity to group 8, and particularly the non-trilate group 8. Essentially, they're selective on small weeds, small seeded weeds particularly. And the larger the soil, the seed size, the less activity there is on that plant. Generally, seedlings that are really, really small are the things that get impacted. So either as they emerge through that treated layer, or in some cases as an early post-emergent application as well. These things move acropedally, but generally not very far from where the exposure takes place. They're lightly bound to soil. They're broken down primarily by soil microbes, and susceptible plants typically don't emerge from the soil. They're moderately persistent, and so dual is a little bit more persistent than peroxysulfone, which is under the names of Focus and Zidua and several others that are combination products within Western Canada. So the symptoms that you see as far as injury with group 15s is that you can get things like buggy whipping that's occurring here. You can end up with leaves that where the margins fuse and you get kind of a tubular type appearance, or onion leafing is how we refer to this as well. Onion leafing can also be leaves that are rolling in on themselves on a longitudinal axis. The thing that you'll see in broadleaf plants is a drawstring effect, where essentially tie a knot in a string and feed it back through the main vein of that leaf and give a tug, and represents more or less the symptom. So essentially that means that main vein fails to expand any further, whereas the leaf tissue in between continues to expand, and you end up with that sort of percolation of leaf tissue up past the vein of the leaf. Group 19 is actually quite interesting. It's an auxin transport inhibitor. So essentially it means that it'll prevent the movement of that natural auxin from the top of the plant down to the rest of the plant, but it also will prevent the movement of synthetic auxins as well. So oftentimes this product gets applied in conjunction with other group 4 herbicides and is a bit of a synergist for those group 4 herbicides. It's sold in combination with dicamba as distinct or overdrive in Western Canada. There's another soil active product called naptalam that is in can be used in turf and vegetable crops. Generally, the diflufenzaper component that's in distinct and overdrive is non-persistent, and the naptalam is slightly persistent. It moves in both the apoplast and the symplast, and it's lightly bound to soil, and it's degraded by microbes in the soil. So the only way that, because we've got products that are in combination with dicamba, the only way that we can really see what the symptoms of the group 19 itself look like is in dicamba tolerant soybeans. And that's what we have here, is distinct applied to dicamba tolerant soybeans. And so what you can see with these is that there's a concentration of symptoms and a little bit of a proliferation of growth here at the nodes. Then you'll have a blank area at the internode areas, and miniaturization of leaves down here at the first node, and it progresses accordingly as you go along the plant. So that's really the symptom that you would expect to see with this class of herbicides. What it does is it essentially concentrates the activity of the that auxin type material. And so in this case, it's simply concentrating its own indole acetic acid activity in the top of the plant. Whereas if these weren't dicamba tolerant soybeans, or they were typically just sort of your standard soybeans, you'd see more of that group 4 impact from the dicamba coming into play here as well. Group 22, photosystem 1 inhibitor, and you'll think that sounds familiar, and it's very similar to the groups 5, 6, and 7s. It's on the same pathway, but a different activity site along that same pathway as the photosystem 2 inhibitors, and very similar types of symptoms, except for there's no selectivity with the group 22s. So you've got rapid membrane disruption from those free electrons that are produced from blocking electron transport, and rapid desiccation as a result. And so you can get desiccation. We've done experiments where we've been doing a trial with Reglone on broadleaf crop as far as desiccation goes. And when we finished the trial, we went back and looked at the leaves of the plant, and they were already starting to exude the cell contents from the surface of the leaf and through the stomata on the leaf. So it works very, very quickly. Within about 15 minutes, you can start getting activities with these things. Paraquat is currently off the market in Canada, and in many European countries, it's also off the market. That one's probably going to fade away into the sunset a little bit, and hopefully we can still retain Reglone or Diquat as a desiccant. They're bound extremely strongly to soils, and there's a component called a binding coefficient. And if we look at the binding coefficient of something like glyphosate, it's somewhere in whereas the binding coefficient for these things is somewhere in the million range. So it's bound very, very strongly, and in some cases it can be nearly irreversible. And so what that means is that the half-life of these things in the soil can be 1,000 days or longer. And essentially it's because the things that are breaking it down, which is either sunlight or microbial activity in soils, they can't get to this stuff because it's bound so tightly essentially between the clay layers in the soil. So once this stuff gets applied, it's there essentially forever. It moves apoplastically. But as a soil active product and with limited selectivity, you really don't get any movement from where it gets applied. So you may get a little bit of diffusion, and you may be able to improve coverage on plants by applying this stuff in the dark. And essentially the idea is that when you apply it in the dark period, you're not getting activation of the membrane disruption component of it, and it allows this stuff to kind of diffuse out from where the droplets have landed on the leaves. And then when the sun comes up the next morning, then it gets activated and all the membranes start bursting and everything starts drying down. So our initial symptoms here, and these are symptoms probably within less than a day on this canola plant. So very, very quickly, so this is at a sublethal dose as well. So you're seeing disruption of the membranes here around the chlorophyll initially, that's resulting in the yellow color because you're losing all the green in the chlorophyll. And then you're also seeing some areas where the actual cell membranes themselves are starting to break down and leak cell contents into the atmosphere. This is a couple days after treatment with chickpea with Diquat. And you can see that if you've got a plant that's fairly large, you can get recovery from buds along the stem with these applications. So it's very important to, particularly when it's being used as a desiccant, you want to make sure that one, you've got really good water coverage. So you're probably looking in that 15 to 20 gallon range for optimal performance of this stuff. So yeah, using low water volumes is not the way to go with these products. And so that way you get good coverage, and you get some of it going into these leaf axils here where these new leaves are starting to form, and maybe kind of getting better impact on some of those. And the other thing that you want to make sure is that if you're putting this stuff on, you're going to be able to harvest it in 5 to 7 days. So because if you wait much longer than that, you will get recovery from these buds taking place. So it's designed, it's intended as a compound for managing harvest and being able to manage your way through short-term weather situations so that you can get the crop in the bin and through the combine reasonably dry. Impact on grasses, very similar. So grasses are a bit of a challenge, particularly when their growing point is below ground. And again, if you get very good coverage on grasses, you can end up essentially starving the rest of the grass plant if you take out enough leaf tissue, and it sets that grass plant back enough that it ends up not recovering from that application. So you can see the change going from the first day to the second day after application. Group 26. So group 26 is essentially group 4s that impact grasses. And the one that is the most well thought of in that respect is quinclorac. So it's the impact on things like barnyard grass and green foxtail with quinclorac. There are a couple of other group 4s that will also work on barnyard grass as well, and holoxafen may fit this class as well because of its activity on barnyard grass. And they're not really sure exactly what the mechanism is in grasses. It's been suggested that it inhibits cell wall biosynthesis, or it's also been suggested that it stimulates the ACCase enzyme and results in production of cyanide that causes the death of the plant. And essentially, the plant will just stop growing and sit there. So the other thing that will happen is that even though it won't kill the oats or wild oats, it does cause sterility in those plants. And so, although not really something that can be used for reduction of competition issues with those weeds, it can help prevent them from going to seed. So that's something to kind of it's something that some researchers may be looking at as a way to manage herbicide resistance in wild oats as well. It's mobile in both the apoplast and the symplast, so it goes both up and down in the plant and absorbed by both roots and shoots. It's fairly mobile in the soil because it's not bound very tightly, and it's degraded by microbes in the soil, and it's moderately persistent. So you can have injury to susceptible plants the year after treatment. So this is what a green foxtail plant looks like that's been treated with quinclorac. The purple is kind of the cyanide impact there, and you can kind of see where that plant has just kind of stopped, and that's essentially what it does. It just sits there like that and doesn't progress any further, but it doesn't really desiccate and die and turn into dust either. It just kind of sits there and does nothing. Group 27, these are the HPPD inhibitors. And so, again, these are carotenoid synthesis inhibitors, which are responsible for the production of pigments in the plant. And so they essentially, the symptom that you're looking for in this case is bleaching, and like totally white, like paper white, the background of my slide white, of the new growth. And eventually the plant will die as a result of exposure to it. The pyrosulfatol can be somewhat soil residual. And so pulse crops like lentils and peas are typically the most sensitive to it. Lentils are not recommended as a recrop option after the use of products that have pyrosulfatol in them. And peas, if you have dry conditions, it's recommended to be very cautious about planting peas following the application of those products. It moves from the bottom of the plant to the top of the plant, so it moves acropedally in the apoplast. And so products in this this class are combination products with pyrosulfatol and bromoxynil. So that's the group 28 and the group 6, and essentially these two are combined because they enhance the activity of one another. So the bromoxynil burns down the plant, and the pyrosulfatol comes in, and they work on the same pathway but in different spots. And so you get some synergy between the two, and you also get the pyrosulfatol coming in and preventing the redevelopment of new tissues that can allow the plant to recover as well. So these are sold under the names of things like Velocity and Infinity, and several other combination products as well. Other things that are in this class are topramazone, so Armazon or Impact. Things that are only in Eastern Canada, things like mesotrione and tembotrione. Some of those are being investigated for Western Canada as well. So essentially the symptoms that you're looking at with these products are bleaching, but the bleaching also occurs at the perimeter of the leaf, and that's showing that it's moving upward in the plant and concentrating at the edges of the leaves from, say, a soil active application, or in this case, a sublethal application. And we're also looking at the older tissues here are the ones that are affected. So you've got the leaves that were being formed where the chlorophyll was being formed right when the application was made are the ones that have turned lily white. The newer ones are kind of developing without much interference. And then you've got the older leaf there that is kind of going down as it has problems repairing itself. And eventually you can get total death of the plant occurring as well. This is the soil residue that I was talking about, and in both pea and lentil. And one thing to kind of keep in mind here is that this leaf curling is not a result of the pyrosulfatol that's in these lentil plants here. There was something else going on in this field, and it kind of highlights some of the challenges with doing things not entirely above board. Again, this producer put pursuit on prior to seeding his lentils, and this curling and stunting effect here on the lentils is actually caused by the pursuit and not by the pyrosulfatol, but the pursuit is accentuating the pyrosulfatol injury and vice versa. So the combination of those two things is resulting in a more severe impact. But here on this pea plant, you can kind of see the how the pyrosulfatol moves in the plant. So these are leaves that were already formed and already had their complement of pigment when the exposure took place, and this would have been a soil residue. So essentially, these were formed at this point when these were starting to form, they got a rainfall. And so he ended up with that pyrosulfatol going down into the roots that had carried over. And so you can see where it's concentrated at the edge of the leaves on these leaves here, a little more so in some of these newer leaves. And as you get closer to the top of the plant and you get into newer leaves, as you have ongoing exposure from the roots, you end up with leaves that are completely white up near the top of the plant. We also have some herbicide lookalikes. things that can resemble herbicides but aren't herbicides. Um, you can get adjuvant injury, particularly with group 1 herbicides, and so the this spot here is a result of injury from an adjuvant, as well as this yellowing that's taking place at the edge of the leaf. And so you can also get like speckling and flecking taking place on these leaves as a result of the burning from the adjuvant. And the real key here is that you'll see it on these older leaves, but very little on the new leaves that develop after the exposure. We've also got a condition called genetic albinism, and what'll happen is that there's a gene mutation in the plant that results in pigments not being produced. And in this case here, it's down in the growing point in a certain point. You can also see it in broadleaf plants occurring. And so what you'll end up with is like one half of a broadleaf leaf will be white and the other half will be green. So it's kind of a neat thing to see if you happen to see it out in the field. So keep your eyes peeled for that. Drought symptoms, as I said, drought and phenoxy injury or group 4 injury on cereal crops are very, very similar, and probably because their root cause is very similar in that the top of the plant is suffering from a lack of moisture. And so symptoms that you'll see there is that lower leaves are starting to senesce, turn brown and senesce. You've got shortened flag leaves up here at the top of the plant. You also have the plant start to take on kind of a blue-gray appearance as the cuticle layer gets thicker. And so as that progresses, you'll get more and more of these lower leaves all dropping off in order to support the growth of this upper part of the plant. And so that's pretty consistent with drought injury. There's also this condition that happens under wet conditions, and it can happen in lentils, it can happen in cereal plants as well, and it's called wet feet edema. And essentially what you've got here is you'll have little pockets, little cells within the leaves where the membranes burst, just because there's so much water going into that plant at one time that you end up with little individual cells bursting and you end up with this little flecked appearance showing up. And you get a very similar symptom like that showing up in grasses as well. Injury from cold weather or a near frost. It's not a hard enough cold snap in order to completely desiccate the leaves in question, but it's enough to yellow them off. And essentially what you've got is ice crystals that are forming in the chloroplasts of the leaves, and they lose their color in the area where the frost has had an impact. And the way to tell when this is happening is that your oldest leaf will be fully involved. Your next oldest leaf may only be involved from point here to the tip. And then your next oldest leaf may only have a little bit of the tip or may not have any symptoms at all. So if you have a situation where you've got 1.5 leaves impacted, and oftentimes what gets fingered in these cases is a group 2 herbicide, but typically this is a cold weather response from the plant, and it's kind of really cold injury. There is a certain percentage of wheat plants and certain varieties that will have natural kinking, and where the heads get caught in the boot, so they can mimic group 4 injury to some degree. But the way to tell the difference here is that it's the concentration of it. When you've got this genetic kinking, it only is maybe 1 out of every 100 or 200 plants in the field, whereas with the phenoxy injury or group 4 injury, you get the thing happening on the majority of plants in the field. Nitrogen deficiency, if you have plants where the bottom leaves of the plant are yellowing, that's a situation where the plant is suffering from a deficiency of either, well, nitrogen or water in that case. And so what it's doing is it's sacrificing the bottom leaves in order to provide that nutrient or water to the top of the plant. And so that's something to kind of keep watch for. If you've got group 2 injury, it'll be your newest growth that is showing your yellowing, and your old growth will remain green. Whereas in these cases here, your older growth is yellowing off and your newer growth is staying green. So that's the way to distinguish between something like nitrogen deficiency um, and group 2 injury. And then we also have sulfur injury that causes leaf cupping. Sulfur injury will come with a very regular cup and very little of the puckering, whereas with group 4s, you'll often get a bunch of puckering, you'll get elongation of the leaf where it's very distorted. This is probably more akin to being confused with group 4 injury. But if it's occurring on the top of a canola plant like this and it's just cupping inward like that, more likely that it's sulfur injury than it is to be herbicide injury. And so that kind of wraps up all the things that I had to talk about here today with respect to herbicides and what they do, how they work, and what they look like when they're working.
Awesome. Thank you, Clark. That was a great presentation. I especially learned a lot. We'll take questions now if you're ready for that, Clark.
Sure.
Perfect. And so as a reminder, if you have a question, please type it into the questions section on the right-hand side of your webinar screen, and we'll get it to Clark for you to answer. First one is, if DIMs are broken down by UV radiation, but we want to spray when plants are actively growing, what time of day should we be spraying?
With DIMs, if you spray into the evening or even in the early morning, it's essentially you're avoiding the high sunlight hours in the middle of the day is what your objective is there. And again, if you're putting these things on at the proper stage, you typically won't see much of an impact of the daylight because you've got ample herbicide there to be able to control it. It's when you start adding up different stresses. So maybe you've got drought, maybe you've got the crop has gotten a little bit beyond the proper stage. You're maybe trying to shave rates a little bit. Maybe your water volume is not super high. That's when you'll start seeing situations where that UV impact starts to take place. But the way to get away from that is to, again, apply first thing in the morning or going into the evening where the UV radiation is very low.
Okay, perfect. Second question is, can you please explain why 2,4-D, MCPA, and dicamba are always talked about in ounce versus milliliter or grams active compared to most other herbicides?
Probably because they're very old, for one. They're older than me and I'm very old. So when these things first started being used, the predominant measure was imperial measurement or US measurement as well. And so things when you talked about weight were in ounces. And that's the thing to kind of keep in mind is that the ounces that we're talking about is a weight measure rather than a volume measure. And that's the challenge with ounces is that you never really know if someone's talking about weight or volume. And so that's why the metric version is much easier to kind of keep that straight because there's no overlap in the terminology that way. But yeah, it's because they're relatively old, and they were one of the first herbicides that were produced in different formulation strengths. And so in order to cross compare, you wanted to be able to assess how much of the active ingredient you were getting on a particular area versus the volume of the product. And so we run into that same kind of thing with the multiple formulation strengths in glyphosate, for example. So in the US, they still refer to pounds active per acre
Hmm.
because
Okay,
they're
good
not
to
on
know.
a metric system.
Sorry. um, Perfect. Are there any resources available that can help with the conversions?
Oh, conversions between ounces and grams.
I'm not sure. It just said conversions. It might mean grams.
Okay.
Yes, it does.
In that case, then the easy conversion between ounces and grams is that there's 28.3 grams in an ounce.
Okay, perfect. Hopefully
And
whoever asked that question heard
I
it.
have a very handy little metric calculator from Sharp that has all of the units in there uh, that I would ever want to use for converting one thing to the other. And there are several, if you've got your smartphone, there are several metric conversion apps on cell phones that are quite effective at converting between many, many different units.
Okay, the next question is quite a mouthful, so if you need me to repeat it, just let me know.
Sure.
With herbicide stacking and seeing potential crop injury the following year, I have heard people refer to herbicide stacking of different products in the same group. I've heard people refer to herbicide stacking of different products in the same group. Is it possible to see stacking the following year from products like flura, fluorozulam and
Flash in them?
yes, and
Okay.
flucarbazone,
Flucarbazone. Yeah.
yeah, applied in the same year? Um, you can answer or an overuse of group 2 products regarding that same scenario. Okay, so answer the first half, I guess, and then I'll go to the second.
Okay. Um, there's been a lot of research that was done back in the early 2000s on stacking, and what really kind of came out of that was that what we see on the prairies isn't necessarily true stacking per se, but what we're seeing is that well, let's use the flucarbazone example for an example. So flucarbazone potential to carry over and impact peas the following year if if you had dry conditions during the year that you applied it. What they found when they did that work, and there was some concern that when you put Odyssey on those peas the following year, that you were getting a combination of injury between the two products, and that's kind of the stacking that we were looking at. In those situations, what they learned from some of the research that they were doing is that there wasn't really enough evidence to suggest that there was stacking going on, but what really was happening from a visual sense was that the plant started expressing injury from the flucarbazone carryover at around the same time that the Odyssey was being applied. And so it's because it took up to about that 4-leaf stage before the peas really started to exhibit injury from 4 to 6-leaf stage before the peas really started to exhibit injury from the flucarbazone in the soil. And that's essentially because that's when all the reserves in the seed ran out for the peas, right? And so it started having to manufacture some of those things on its own using those essential amino acids. And so that was right around the same time that the Odyssey was being applied. And so there's some implication that the reason that you were getting the flash was because of a combination of the two of those. But what they really found when they had small plot areas in the field is that they could have just the flucarbazone in one plot showing the same symptoms as the flucarbazone plus the Odyssey in the next plot. And so they weren't really seeing what you could say was true stacking in those situations, but more just the expression of the flucarbazone injury and the timing of that injury, so to speak. Now, the question here is that if we're putting on a somewhat soil active burn-off product in the fluoroacelam, could you see injury from the two of those? I guess it's entirely possible. But again, it's likely going to be an accumulation of circumstances that causes that situation, where you would have had to have had dry conditions the year before with the flucarbazone, and then you'd also have to have cool conditions that gave an impact of with the fluoroacelam. So there's lots of things that have to really line up for that to happen. And so it's going to be a very, very, very rare situation where you end up with that type of circumstance taking place.
Okay, I think you've actually touched on the second half of the question, but I'll read it to you and then you can tell me if
Sure.
you've answered it so far. So it's an overuse of group 2 products regarding the same scenario with those 2 other products, but with an
Mhm.
addition of an IMI the following year. Will these 3 products stack up and cause crop damage, or is it not possible because there are different subgroups of group 2?
Um, if well, where you may see that issue taking place, if you've got the thing that I'm envisioning is that the only place where you would see those things all stacking up together would be prior to an IMI-tolerant wheat, where you would have maybe flucarbazone applied the previous year. Then you've got, and there's a set of products out there that have a combination of flucarbazone, fluoroacelam, and carfentrazone all in the same product that get applied as a pre-seed burn down product with glyphosate. And then there's some sustained soil activity from particularly the flucarbazone component to be able to suppress wild oats and some of the broadleaves in that field, right? But then the only kind of situation that I could see there being an IMI involved in that route as well would be if it was an IMI tolerant wheat. The other thing that other scenario I could see where that might come about is if you had something like an Odyssey application the year before, and then you put one of those combination flucarbazone fluoroacelam type products on in the spring, and you had a big gusher of a rain and cool conditions when the crop was coming up out of the ground, and the year that the Odyssey got applied, you ended up with dry conditions that year as well. And so, yeah, again, it's one of those things where you'd have to have a lot of things line up in order for that to really present in the field. It's not impossible, but it's not very likely either.
Okay. How many ounces per acre do you consider low rates of MCPA to use at the 2-leaf stage in cereals?
Generally, what we're looking at there is 4 ounces active, or 280 grams active per acre or lower, and that allows that MCPA to be used down to that 2-leaf stage. If you start getting up into the higher leaves, the higher rates up into the 8-ounce, 6 and 8-ounce range, you probably want to back it off another leaf to the 3-leaf stage.
Okay. Is there a resource or website where we can view herbicide injury photos?
Oh, Oh, there are some resources that are available. I believe it's the University of Minnesota has some injury symptoms on cereals, I believe. And they've got a publication it's a PDF version of a publication that has that. But we don't have any of those resources ourselves. The handbook for our crop diagnostic school does have some of those resources in it and available. So that's one source, but online, no.
Okay. This individual would like you to show the wet lentil environmental slide again, please.
The wet, the okay. Let's go back here. The wet feet edema,
Yeah,
that
must
one
be.
there.
Yeah, they just wanted to see it again. Then you can answer this next question while they get a chance to look at it.
Okay.
Is there any truth behind there being some residual weed control with 2,4-D?
Um, a lot of that depends on the rate that's used. 2,4-D is largely completely gone from the system within 3 weeks to a month, under ideal conditions. So the amount of activity you're going to get out of 2,4-D, and the other thing to kind of keep in mind is that, and I'll kind of try and draw this with my little pointer thing. Here, maybe I can do a pen here. Yeah, why don't I do a pen, and I'll do it right on this picture here. So when you're getting herbicide breakdown taking place, it kind of progresses on a curve that goes like this. And I got to make it so that my mouse pad doesn't fall off of my tray here. And it goes like that. So essentially, it breaks down fastest right away after application. And as you get further out in time, the breakdown slows over time. And you're getting down near the x-axis at this point of your curve. So once it gets down to the point where there's not a whole lot of it left in the soil, the breakdown occurs slowly. And this is either referred to as an elliptical curve, or sometimes it's referred to as a biphasic breakdown curve, where it breaks down very quickly right out of the gate, and then it breaks down very slowly after that. So with 2,4-D, when you're looking at this spot out here on the curve, only being 3 weeks to a month away, and so you're looking at maybe you're going to get 3 or 4 days where you might get activity on seedling weeds coming out of the ground during that stage. The other time application window where you might get some soil residual activity out of it is if you put it on in the late fall when it's really, really cold. And what that does is that essentially suspends the breakdown. And what you may get is early in the spring, you'll get some impact on emerging winter annual weeds at that point, just as the snow is kind of melting and clearing off, and you get some of those winter annual weeds starting to push through that really cold soil. You'll get some activity out of the 2,4-D in the soil that way. But once you're into the heat of the growing season, it breaks down so quickly that you're really not going to get much in the way of activity out of it.
Okay, you've actually touched on the next question as well. You keep doing that to me, you keep tripping me up, but um, you're just ahead of yourself. That's good.
Sure.
We've heard, so the question is, we've heard different companies claim 2,4-D will break down approximately 1 ounce per month over winter. Do you have any research or comments on that? But I think you've kind of touched
Um,
on that.
do I have research at my fingertips? No. Um, do I have an understanding of what's required for herbicide breakdown? You bet. Because for herbicide breakdown, you need two things. Well, you need three things. First, you need the herbicide. Two, you need moisture in order for that to happen. And three, you need warm temperatures for it to happen. And so you don't have that third thing during the winter months. You don't have the temperature. And so essentially everything stops over winter. And as you get a warming in the spring, you'll start to get a little bit of activity. But because your microbial populations are at a fairly low ebb as they're coming out of winter, you don't have those really robust populations that are going to be very active at breaking that material down right away. And so it's going to take one, some time because it's cool, and two, some time because you don't have really big populations yet, and it's going to take a little time for those populations to grow to the point where they're really robust at breaking down herbicides. And so when we talk about herbicide carryover, that's why we look at rainfall from the 1st of June to the 1st of September as kind of our window to kind of gauge what the risk of unexpected carryover is going to be. And it's because that early portion of the year, that April, May timeframe is when the soils are really cool and your microbial population really hasn't kicked into that 100 miles an hour kind of activity yet. And so from that perspective, you could get some extension of the residual activity of the 2,4-D in that case.
Next one for you is I had to look at a lot of fields last year for issues with plants. It ended up being drift when applications were being done under extremely windy conditions in 2020. Are there regulations as to when herbicides can be applied?
Are there regulations? Yes, and it's a bit of a joint responsibility between the province and the federal government. And so what you'll do is find on individual labels wind speeds that are recommended as far as maximum wind speeds, and they're not as good at minimum wind speeds, unfortunately, which sometimes can be a bigger issue than maximum wind speeds. When you've got windy conditions, what'll happen is that if you've got fines that are prone to drift, they'll get caught up in that turbulence and they'll kind of get shuffled around and diluted up in the atmosphere and evaporate and disappear that way. Where the bigger challenge for more extensive drift takes place is if you've got dead calm conditions, you go through with the sprayer, you've got fines, and what they'll do is they'll just hang. And in those situations, you often have very high humidities as well. And so those small droplets just sit there and they're suspended. They're just hanging in midair and not really falling because they don't have enough weight in order to fall. And then as it generally, this is something that happens very, very early in the morning. And as the sun comes up and you start getting more of those daytime winds starting to kick in, that concentration of either those really, really fine droplets, or in some situations where you've got relatively volatile compounds, things like dicamba and 2,4-D esters and things like that, can concentrate under what's called the inversion layer. And then once that wind starts coming up during the day, sometimes it has a little bit of time when it figures out what direction it wants to go eventually. So you can have it kind of sliding around the landscape a little bit, and then eventually it'll kind of move off in the direction that the wind is going to be progressing that day. And that whole mass will slide across the landscape, and sometimes it'll flow down drainage pathways and things like that. And that's where you get more extensive drift injury is from some of those inversion or vapor type drift scenarios. And that's why with those volatile type things, or if you've got nozzles that tend to produce a lot of fines, that's why it's risky in those situations applying early on in the day versus in the versus midday where you've got breezes taking place. I don't know if that answers the question or not, but
No,
I
I think
think,
he did hit it
okay,
right on the head with that one. Yeah.
okay.
Next one is certitude was just registered for canola. Do you think there's any chance there will be group 27 damage on canola with this mix?
I have no idea.
Okay, good to know.
I have yet to lay eyes on certitude, so BSF hasn't invited me out to see their plots.
All right. Okay, so this is the next one's your last one, and then you can.
Hint, BSF. I'm looking to see some.
Alrighty, so this is your last question. She's a big one again too. Regarding resistance in wild oats, there seems to be less information on group 2 versus group 1 resistance. Group 1s are treated as a herbicide group with 3 subgroups, while group 2 is generally referred to as a single group. Wild oats resistance to the subgroup FOPS can often be controlled with DENS. Do you find the same goes for group 2, where IMI resistant wild oats could be controlled with TZP or SACT? Would rotating subgroups of group, although not ideal, in group 2 products would help reduce the incidence of resistance? Ooh, tough one for you.
Yeah, I think that's a pretty risky strategy, and in part it's because of the number of mutations that can be produced in a plant that one result in a viable plant and result in group 2 resistance. When we look at the frequency of mutations to particular groups, group 2s have the highest frequency of viable mutations within plants typically. And the number that gets thrown around is 1 in 100,000 plants will have a viable mutation that will result in a group 2 resistance. In a lot of cases, there is very strong cross resistance across all the subgroups within group 2. That being said, that it isn't necessarily universal, and it's going to be species dependent as to how much cross resistance there is. But I think that's a very risky strategy to try and approach something where you know there's resistance to group 2, or the potential, let's put it this way, the potential for resistance to group 2. And there's some of those mutations that result in resistance to both. So that's the thing, is that you never know when you're rolling the dice which mutation you're going to end up with. You could end up with the one that results in resistance to the IMIs. You could end up with the one that's resistance to the TZPs or the SUs or all of them. And whatever one has the resistance to the most herbicides is going to be the one that predominates.
All right, perfect. Well, that's all the questions they have for you. So I'd like to extend a great thank you to you, Clark, for your excellent presentation and excellent Q&A. And I hope you have a great rest of your day. And I would like to thank everybody for joining us today for the webinar and listening to Clark talk. If you've entered your CCA number during registration, we'll make sure to send that along to the appropriate person so you receive credit. Credits are only available to those who watch the webinar live, and you can easily find recordings of our previous webinars by visiting our website and searching for webinars. And more information on the topics mentioned during the webinar can be found at www.saskatchewan.ca/crops. And if anybody has any questions regarding this topic or anything else related to farming, please contact your local Sask Ag regional office or the AKC at 1-866-457-2377, and we'll be sure to put you in contact with a specialist to help answer your questions. Thanks again and have a great day.